All right, so I think everyone is starting to come in. So without further ado, let's get this webinar started. So thank you everyone for joining us today for our webinar in predicting clinical efficacy with Alzheimer's disease iPSC panels, presented by Dr. Nadia Eckert. Just to let everyone know ahead of time, there will be a Q&A session at the end of the webinar. So if you do have questions during this time, we recommend you to actually submit your questions in two formats, either in the comment section, which should be on your right at any point during the webinar. If you are using StreamYard, you'll see that there's a button as shown in the first left box that you would click on. Or if you're using LinkedIn, the comments button as shown to the right of that box. If you'd like to send over questions privately, we also have an email that you can send the questions to, which is questions@elixirgenside.com. And just last point, for any questions that aren't answered during the webinar, we will send emails back with your answers. So please, when you're using your name and email, make sure that it is one that you can receive emails from. And with that, let me introduce our speaker, Dr. Nadia Eckert. She received her PhD in biochemistry, cancer biology from Drexel University School of Medicine. She also completed her postdoctoral fellowship at the Wistar Institute. Before joining Elixirgen, she also was the associate director at GSK, working on sequencing, gene editing, target identification validation, and immunology. And currently, she is the senior director of research and development here at Elixirgen Scientific, where her team develops services, including focus of today's the Alzheimer's disease panel. Without further ado, Dr. Nadia Eckert.
So today I get a chance to tell you all about our efforts to build a cell model for late onset Alzheimer's disease, and it's based on iPSCs derived from Alzheimer's patients. I'm suspecting this audience, you're probably most familiar with studies and cell models of familial Alzheimer's disease. And however, the 95% of all Alzheimer's cases are actually late onset, and they start with patients being in their mid to late 60s. So the symptoms are generally actually the same, involving memory loss and thought and language difficulties due to loss of synaptic connections. With spontaneous AD as actually not due to any mutations, specifically in either APP or xenolins, but it's considered a polygenic disease with a handful of known risk factors, such as ApoE4. And with molecular characteristics involving the same characteristics of the familial disease, such as amyloid plaques and phospho-tau tangles formation, which eventually, as you know, block neuronal transport systems, resulting in loss of synaptic connections. So I will start with describing our proprietary platform first, how to differentiate iPSCs, how we differentiate iPSCs, and then talk about using the AD patient iPSC-derived excitatory neurons to model AD. And besides the neurons, we also develop microglia and astrocytes with the goal to put together a triculture model. And I will show you some data on this as well. Right. So there are actually two different ways to differentiate iPSCs. One is the lengthy one by using retinoids and growth factors in certain compounds, which takes months. We develop a quicker path, which starts with expressing transcription factors to push these either embryonic stem cells or human iPSCs into a certain lineage. And it was published by our founder, Dr. Minako, in 2020 Cell Reports paper. He took 714 transcription factors and introduced them to embryonic stem cells, and then carefully analyzed expression profiles that these transcription factors drove in these embryonic stem cells and matched them up with organs and tissues. And from that analysis, the human gene correlation matrix was created, which allows us now to identify which transcription factors contribute to differentiation into which specific cell types. And this became a proprietary platform for Lexogen that we use to differentiate iPSCs, either patient-derived or from normal controls. So for us, actually, to build an efficient and predictive iPSC-based models, we also take into account the wealth of knowledge out there from a variety of sources, and actually our home-built databases, which includes genomics, transcriptomics, the RNA-seq in bulk or single-cell RNA-seq, and also proteomics data, either from patient cells or tissues and from healthy individuals. So this all allows us to ensure that when we build cell-based models for anything Alzheimer's or any other disease, we create it to be as disease relevant as possible, with recapitulating all of the pathology and expressing the relevant biomarkers with these cells being functional. So, and typically to achieve a cell type of interest, that's the next slide. We start by selecting the transcription factors using the gene correlation matrix, which I just described you. And we then go through rounds of culture and seeding density optimizations, and then finally arrive to the cell type that we need. This is followed up by extensive QC and morphological analysis and functional analysis, actually, and finished by creating manufacturing processes and SOPs to make sure that we can produce quality iPSC-derived cell models for drug testing, MOA research, and we can produce them also in large quantities if we need to. So this technology has proven to be incredibly fast. It only takes 7 to 10 days from an iPSC, expanded iPSCs, to fully differentiated neuron, which neurons are mostly the cell types that we work with, actually. So the source of iPSCs seem to be kind of irrelevant for the success of differentiation. We actually took iPSCs from different sources and differentiated into excitatory neurons. And we compared the batches of excitatory neurons differentiated from same iPSC on different times. And the expression profiles happen to be very similar. So it's a very it's a process, it's a very dependable process, I should say. So besides QCs for the markers, we also, as I said, run functional assays. And typically for the neurons, it includes microelectrode arrays, phagocytosis for microglia, the dopaminergic neurons get profiled for dopamine release and so forth. So and on top of that, we have capability to expand our stock into billions of cells. And this is actually done on the Japanese side of Alexogen Scientific at the Alexogen Scientific CDMO. So the next slide has all the variety of this iPSC differentiated cells that we are now able to produce, which includes many types of neurons, as you can see. Microglia, astrocytes, and skeletal muscle cells. And we're currently almost done developing oligodendrocytes, which was a lot of fun. We have some cardiomyocyte development work going on, and NK and T cell development work. So if you're interested in any of that, you just send an email to Henry, and he'll help you connect with us and talk about your project. So yeah, developing cell types is a lot of fun always, but we also want to put them to use to make them serve the research community. So last year we released our AD panel, which is essentially a collection of patient iPSC-derived excitatory neurons, and also isogenic microglia and astrocytes. So we have been extensively characterizing the cells by whole exome sequencing, RNA-seq, and testing a few well-characterized compounds. And those that aim to decrease AD biomarkers in the system, such as amyloid 42 and 40, amyloid beta 42 and 40, and pTau. So, if you've been testing compounds in the normal lines, you might be thinking, why bother with patient iPSCs, right? So our overall goal here is to set up a cell-based system that not only reflects the pathology of Alzheimer's disease, which it should, but also carries the genetic makeup of the patients, and therefore can answer some very important questions. For instance, we might be able to use this model to figure out the genetic determinants of drug sensitivity or drug resistance. That would be very handy to know this information prior to the beginning of clinical studies. Or we can help you hone in onto a defined mechanism of action, maybe better than you could have done it in the normal cells. So we source our cells from the California Institute of Regenerative Medicine, and they have over 1,500 iPSCs from patients with a variety of conditions, including over 60 iPSCs from patients with Alzheimer's, plus a larger number of matching controls. And when it comes to sorting iPSCs for Alzheimer's or any other neurodegenerative disease, there are other sources as well. So we keep on growing our collection here. And so far, in the next slide, we obtained about 20 for the first kind of rollout and tryout of this model and the building of this model. We obtained about 20 AD iPSCs to start with, and plus some healthy controls. So, and we differentiated them all into, as I said, excitatory neurons and astrocytes and microglia. So there's pretty much all of them have been differentiated to excitatory neurons, and we have probably around 6 pairs of excitatory and astrocytes, and some pairs of complete sets of astrocytes, microglia, and excitatory neurons. So we have the medical histories on all of these patients, including ApoE status, sex, race, sampling age, age of onset, height, weight, and so on. And we sequence all of these iPSCs, so there's info on gene variants and mutations for every line that we have. So, I mean, our hopes are, and this set's going to be very useful to the research community, and there are many ways that we can take patient genetics into account when we do our experiments. So we first produced a batch of 7 lines of ED patient-derived excitatory neurons, and we had 1 line from a healthy control subject. So here's how they look at week 4 post-differentiation. And the neurites are stained with tubulin beta 3 in green, and we glued 1 marks up in neuronal bodies in red. So the differences in morphology, and marker distribution in AD versus control is not so pronounced at this stage because at week 4, these neurons are relatively young. However, the more they age and the more we notice the differences, and our colleague, Dr. Lin from RICO, has published some data on differences that she observed in week 10, 10-week-old neurons. So these differences include more loss of synaptic connections in AD lines versus normal, and less neural bursts from AD lines versus normal. And of course, this is published data, so you can go ahead and read about this. So we are able at our site to maintain the cells up to week 12 so far, and they're relatively healthy at that point. We haven't tried carrying them longer, but I suspect that's actually possible. So before I continue describing our cell model, I would like to delineate the protocol that we're following so that you understand experimental flow. So we start with iPSCs, right? So this is our overall plan that we have for this model. And we want to hit all the points, because if we don't, then it might not be useful as a model of Alzheimer's disease, right? So what are we looking for, right? We're looking for amyloid accumulation in cell cultures. We're looking for secretion of amyloid beta 42 and 40 peptides, an elevated ratio of amyloid beta 42 over 40. And we're looking for activation of glycogen synthase 3 beta, which phosphorylates and hyperphosphorylates phospho-tau. So we're also looking for higher levels of phospho-tau phosphorylation in the cells. And we're looking for higher levels of phospho-tau to tau ratio in AD lines versus normal. And later on, so this is something that we can only study in the mature neurons, but we are planning to do the studies. We will culture the cells to week 10, and we'll be able to assess loss of synapses and decrease of synaptic plasticity in these neurons and altered electrophysiological activity, something that Dr. Lin has established in just a few AD lines versus normal, but we plan to expand on this study. And also the AD model is supposed to have, the AD panel is supposed to have increased oxidative stress and reactive oxygen species levels, which is something we are still examining. So next slide. So, and I thought, so let's take a look at the experimental flow of this protocol. And what we do here, we start with iPSCs, and we expand them to before we transfect them with transcription factor cocktail. And that's as neurogenesis begins, these cells reach a neural progenitor stage, and they do this super fast, actually, at day 3 to 4. So at this point, we freeze them into multiple aliquot at day 3, and then we thaw an aliquot, and we proceed we culture these cells to day 10 and do QC. So at that point, they're supposed to be expressing all the markers that I've just shown you, such as VGLUT1 and tubulin beta 3 for excitatory neurons, for example. And when QC passes, we thaw these day 3 cells, and we allow them to differentiate to into either 4 or 6-week-old neurons. So we wait for 42 days from that point forward. And then we treat them with drugs, various concentrations of drugs to build those response curves. So, well, I'm not showing you all the little details and the data that we spent actually months figuring out how long we need to culture these neurons to start registering levels, decent levels of phospho-tau, tau, and amyloid beta 40 and beta 42. So, and the answer is that you have to wait until at least until week 4 minimum for tau and week 6 minimum for amyloids. So we profiled amyloid beta 42, beta 40, tau, and phospho-tau. So 40 is not included here, but we did it in the 7 AD lines of excitatory neurons, and we found out that AD lines express significant levels of all these biomarkers. This is what we were aiming for. And there's some always donor variability across the board. And that's, if you work with any primary cells, you're going to have donor variability. And to the degree that you can deal with that variability can make actually a big difference in whether your target validation project will go forward or get tossed. So, so here, what we did, we took a ratio of phospho-tau to tau, and it seemed to normalize the levels of phospho-tau to tau in all the AD lines. And I know that the sample size is small, so the ApoE heterozygotes of 3 to 4 are in black here, and ApoE homozygotes by ApoE4 homozygotes are in blue. We do not really observe any significant differences between the two. And maybe there is a bigger difference if we're comparing the ApoE2 versus ApoE4, but we haven't done this yet. So we keep differentiating more and more more and more iPSC excitatory neurons, neuronal lines to be able to do this analysis. All right, so As I said, these cells come from a patient, right? And they differentiated in the cell type of interest. So this is something that we can typically follow. A typical experimental work plan consists of a drug treatment, such as in our case here, BACE1 inhibitor, GSD3 beta inhibitor, and that aims to reduce either amyloid beta or phospho-tau, right? So we followed it up with in vitro assays or omics data collection, which allows us to have a better understanding of which genetic determinants of resistance or sensitivity to the drug are important, and also have a bit of a better idea of the drug's MOA, depending on the goal of the experiment. So we actually went back, as the next slide, to re-examine the expression of amyloid beta 40 and 42. And now we have 7 AD lines and 4 normal healthy lines. And when I say healthy, it's not real, like these patients do not have Alzheimer's. They may have other conditions as well, because they are aged matched, and at 70 years old, they're probably almost guaranteed to have some sort of health issue. So just like in the patient, so this are 7 AD and 4 healthy lines, excitatory neurons at week 6 post-thaw. And just like in the patient's brain tissues, amyloid beta 4 is a lot more abundant than amyloid beta 42, but all lines express both, and most AD lines express more amyloid beta 42 here than what healthy lines do. So let's go to the next slide. And we calculated ratio of amyloid beta 42 to amyloid beta 40, and it provides a more accurate indication of Alzheimer's disease pathology. If you familiar with publications of familial AD, you'll know that it's a hallmark of Alzheimer's disease pathology in these cell-based models. So what we observed here, so on the right, we did a t-test analysis, and we observed that the AD patient-derived iPSC neurons, on average, have the ratio of amyloid beta 42 to 40 is 2 to 3 times higher than the healthy controls. So, and this result has been statistically significant. So, well, we took the cells and we treated them with BACE1 inhibitor, lambecestat, for 72 hours at week 6, and we built these dose response curves. So we know the drug works in these cultures. We also found almost all of these lines have very similar IC50s and kind of the same range, and with one line being an outlier, we thought it was an interesting observation. And we thought that if we were to do statistical analysis on this, we'll have to have more lines for this. But at the same time, in parallel, next slide, we were examining phospho-tau and tau in the same cultures. So we are measuring amyloids, beta and 42 and 40 in supernatants, and we use the lysates of cells for tau and phospho-tau studies. So here, and we keep expanding, adding more and more lines to this. So my team actually differentiated this 4 healthy control subject lines and 10 AD patient-derived lines to excitatory neurons. Again, they're reaching in week 6. And then, well, at week 4, it makes sense to measure phospho-tau, but we wait a little longer, and we also take advantage of kind of using supernatants for the amyloid ELISAs and lysates for the phospho-tau ELISAs. So in parallel to the amyloids, we have profiled tau and phospho-tau in the cells, and of course, that we find always high variability in patient-derived lines, as I said, so in both total tau and phospho-tau levels. And next slide. Yeah, and I can also say, so I will talk about the ratio of the phospho-tau to tau in the next slide. But for now, I'd just like to say that We can also, by the way, detect phospho-tau by high content imaging. This is one of the assays that we're consistently running. So the GSK3 beta inhibitor normally decreases phospho-tau phosphorylation. As you can see here, actually happening by decreasing the intensity of phospho-tau tyrosine 231 in both soma and neurites, and it inversely correlates with the intensity inversely correlates with the GSK3 beta inhibitor concentration. So we can actually use ELISAs and we can use high content images, but most of the data I'll be showing you comes from ELISAs, but that's just an option. All right, next slide. So in familiar Alzheimer's cell models, the phospho-tau to tau ratios is typically significantly elevated compared to healthy cells. And it becomes a key characteristic of Alzheimer's disease pathology. It also helps us to normalize expression and data in this case, since we're experiencing this patient-derived cell variability, which is very common. So, and it's also often used, the phospho-tau to tau ratio is often used as a marker to study disease progression and drug responses in cell models. So what we did, we calculated the phospho-tau to tau ratio here. Again, it reduces the variability from individual differences in total protein levels. And overall, you can see that 9 out of 10 AD lines had a ratio of phospho-tau to tau 2 to 4 times higher than the healthy controls. And again, we did the next slide. In the next slide, proceeded with the t-test and found out that as a group, the AD lines actually have statistically significant higher phospho-tau to tau ratio is expected, from the previous slide, but also it helps us. It builds our confidence in what we're seeing as a true useful AD model, very comparable to what's observed in the familial AD models as well. So we treated these 4 healthy lines and already 11 AD lines here with GSK3 beta inhibitor, and we have measured phospho-tau by ELISA. So all 4 normal lines showed a similar profile when it comes to the drug response, while in the AD patient-derived group, we have this much higher variability where the 2 lines have been very sensitive to the drug, 2 to 3 lines are kind of moderately sensitive and mid-range, and the rest are comparable to the control lines, the healthy lines. So we have collected some exome sequencing data, as I said in the beginning, and for all of these lines, and we were able to compare responders versus non-responders and use some statistical analysis to identify variants, genes, and signaling pathways responsible for this drug sensitivity. So it has the final goal to help with patient stratifications, but I will speak to both advantages and disadvantages of this method. Next slide. So the caveat here that we have a limited number of samples, and we obviously would not be able to conduct genome-wide association studies, but we were able to use Cohen-D's method to answer the question and hypothesis whether any of these 29 related genes that have variants that are distinctly different in sensitive lines versus normal response lines. And by using this 29 AD-related genes, I'm talking about the risk factor genes for Alzheimer's. Okay. So we found that about 8 to 10 variants. We found that 8 to 10 variants that are on the top, and they're statistically significant here with the Cohen-D analysis. And we thought that the next step would be to validate these results. And the question that we can ask is, if modulating the genes or individual variants, changing excitatory neuron sensitivity to GSK3 beta. Okay, so, and this technique can be applied to pretty much any drug. Doesn't have to be GSP3 beta. But as I mentioned, the statistical rigor can be supported either by increasing the number of lines that you're analyzing or coming up with a very defined hypothesis. Small number of genes, but that these genes might be responsible for the variants of these genes might be responsible for the sensitivity of the drug. And we are actually continue working on this experiment. And as I said, we're adding more and more lines in our collection. So as I mentioned before, we have we can differentiate isogenic neurons, astrocytes, and microglia. Pretty much all of these iPSCs that we received from CERN. And what do you do with those? You build a triculture model of neurons, astrocytes, and microglia to recapitulate the exact architecture that we can find in Alzheimer's brain and see if the triculture models are actually better, worse, same. How are they more predictive than excitatory neuron-based models? So all of these questions is something that we're trying to answer at the time. So next slide. So the astrocytes are actually the longest. So it takes about 42 days to differentiate them to functional astrocytes. But here they are, and they're expressing the markers that they're supposed to be expressing, such as GFAP, CD49F, and ALT-H101. And AD-derived astrocytes overall tend to acquire more aberrant and elongated morphology. It's what we observed. So we have several of those lines already made, and we are ready to conduct some of the triculture experiments on AD lines versus healthy. So another cell types, the next slide, is a cell type that we have is microglia. And our microglia expresses IBA1 and TMAM119 in both healthy and AD lines. Can just comment on the morphology because we have just a limited number of lines, so we'll assess that later. But we are ramping up the differentiations, and we just have some microglia AD lines available at the moment. But while we are ramping up the production differentiation of various microglial lines, we wanted next slides, please. We are looking to assess how functional our microglia is post-differentiation. And what we do is we incubate them with Frodo-stain zymozyme particles. And Frodo is a pH-sensitive dye that only fluoresces when being phagocytosed by microglia, microglia, macrophages. So we're testing microglia here. So you can see that phagocytosis begins rapidly within 12 hours after adding the Frodo particles. And by 48 hours, most microglia cells have Frodo dye in lysosomes. We continue to characterize these cells. So first we did QC for all the appropriate markers. You can see the expression of CD45, which is for all the immune cells, and IF1 and TREM2. So we also treated them with either TNF-alpha or LPS to upregulate pro-inflammatory cytokines, or and you can see that that's happening. That's the data on the right. And if we treat them with IL-4 and IL-10, this microglia is shifting to M2, as it should. So next slide. So here you can see that what we put together in the past few months is all the cell types, and we figured out how to have these the astrocytes, microglia, and neurons on the same plate, happily living together, which is not an easy task. So you can see that now they're all good. Neurons expressing tubulin beta 3 here in purple, astrocytes expressing GFAP here, and microglia is carrying GFP marker. So what we're doing now in our group is we're conducting some neuroinflammatory assays on these cells on just a healthy line at first. And as soon as we get these results, I will be thrilled to share it with you. Well, here they are, and thanks to everyone at Deliverables, there's been a lot of support for building out this AD model, and specifically, I'd like to thank Yaseman and Zabi, Katuska, Titus Rodriguez, Chelsea Myers, Moises Rodriguez, for working extra hard on the AD model, and Abby for working extra hard on developing Microglia. They very much appreciated all your efforts. And a spoonful of good luck to them. So I think we can go on to questions.
Awesome. Thank you so much, Nadia, for going through that great data and talking. I know we've gotten a couple of good questions in the comment section, as well as a couple sent in via email. So yeah, just once again, if you do have additional questions you'd like to ask, please use the comment section, or you can send your questions if you don't want to necessarily share in the comments to questions@electrogensci.com as shown below. So I think we can start off with one of the questions from the comment section. We had a question in regards to the changes in the cells. Have we been able to measure some of these changes between the healthy and the Alzheimer's disease lines for glucose metabolism, autophagy, and/or mitochondrial function?
All right. Well, thank you for all these ideas, and I would just say not yet. Typically, we apply mitochondrial assays to ALS models, but we haven't tried this yet, and I think it's a very interesting idea. The glucose metabolism, and what else was there?
Mitochondrial function.
But the autophagy, we haven't tested. We haven't had any assays done on it.
But these are assays that we would feel confident being able to try if requested, correct?
Yeah. So for the metabolism, there's a blank on the name. Yeah, but we've ran a couple of metabolism assays in the past. With the autophagy, we haven't done it yet. If there's a commercial kit available to profile autophagy in cells, what we do normally is we have a couple of weeks of assay development to see if that works well in our cell types. And then typically our clients request to test the drugs and see how it affects autophagy in our system. Or any other process, I should say.
Okay. Yeah, and I know if there are other particular types of ads that I know we'd be happy to discuss.
Right.
Okay. So for one of the email questions, I think I got here is Electrogen looking to expand into other disease types other than Alzheimer's disease?
Yeah, so not from the same iPSC, right? So so we have access to ALS models, Parkinson's disease, and rare diseases like Rett syndrome. Modeling of diseases is very specific. So, but I think we're very encouraged by our success with the AD model. So we're definitely working on the AD model at the moment, and we'll be expanding to other diseases as well.
Gotcha. And to everyone on here, if that is of interest, please feel free to email us, and we'd be happy to discuss some of those other diseases as well towards this type of model. I think we have another question from the comments. So from the pTau total tau ratio, they're curious why the APOE 3-4 ratio uh, shows to be higher than the APOE 4-4 ratio, and as well, the APOE 4-4 response seems closer to the APOE 2-2, and why do you think that is, and do you have a particular hypothesis about it?
My hypothesis is we don't have enough lines to convincingly say that we observe statistically significant differences between the alleles.
Gotcha. So
But it's a thank you. It's a nice hypothesis to test. It just needs to be tested on a lot more lines.
just so really the increase in the different patient lines that we're bringing in now could potentially help,
especially more of APOE2 alleles, because a lot of patient-derived lines are focused on APOE4 carriers.
I see.
So it's kind of a APOE carrier heavy. if you notice in iPSC sources.
I gotcha. So definitely opening up to the different type of patient lines will definitely help assist there. So for another question that was emailed in, other than we did show the drugs with the GSK3 beta inhibitor and the BACE1 inhibitor. Are there other drugs that Electrogen is planning to test in this model?
Oh boy, do I have a list. Lecanemab, right? Lecanemab. Would like to profile M19 and see if we can modulate an NMDA receptor, right? And also the other ones, class of drugs is the cholinesterase inhibitors. So we hope to profile them with the help of our clients. So if you have a compound that you'd like to test in these models, I think we'd love to do that.
Okay. Yeah, I agree there. If anybody does have particular compounds or does they would like to test, we're very happy to discuss being able to run them through in this in our platform. Another question that has come in from the emails, do they need any licenses to run the AD panel?
To assay the AD panel, actually, question for you, right?
Yep, I see. No, that is a good question. For the person they emailed in, you actually don't need a license for us to run these AD panels. Elixirgen actually has the license for these lines as well as for many of the tests that are being done. So all the data generated, all the lines fall under Elixirgen, but all data and such generated would be yours and yours alone. There's no additional license needed. Right. And then just making sure. Another question in the chat. For the triple or for the triculture, can you explain if we have specific ratios, and of those ratios, is the ratio and sequence important? And then what could we state what we have used?
I suppose you're referring to the sequence of addition of cells and how we manage all the cells, and the answer is yes. So because you ideally want to culture neurons and astrocytes. Well, first of all, culture neurons and astrocytes together is way better for neurons. They feel better and healthier. So these guys go together first, and Microglia is an add-on after the neurons and astrocytes have been in culture for some time. And we have a protocol developed, so if you want to work with us in this Tricultures, you're more than welcome to reach out, and we'll discuss the project.
Yeah, so definitely reiterating what Nadia has mentioned. If you are interested in generating these tricultures, we're happy to discuss and walk through protocols and such on there. Right, I think we have time, for two more quick questions. one that was emailed in is, thank you for sharing the need or for the number of lines that have been run through, but can additional lines be added?
Absolutely. And that's what we're doing actually. So, and if your project involves more than 15 or 16 lines at the time, well, actually we have differentiated for more recently, so 20 lines now. Then what we do is, since our differentiation protocol for excitatory neurons is so dependable, we can order up more for your project, and you don't actually have to be married to CERM stock. You know, if you have a source of them, iPSCs from AD patient from somewhere else, that's also fine as long as licensing is good. So, I mean, we can, in theory, differentiate hundreds of lines.
Mm-hmm. Yeah, that is correct. Yeah, no, that's a great point to bring up, Nadia. We have shared all the lines that we have access to, but if anybody has iPSCs source that they would like to send over to us, we can definitely differentiate and work with those as well. Then I think one of the last questions emailed in is so a group asked, what type of functional assays have been planned for the Alzheimer's disease lines, and have there been any plan for the Triculture models?
So one of the priority assays is our neuroinflammatory model. So for our Triculture, we're going to be treating these cells with either LPS or soluble amyloid beta, and profiling cytokine release from either microglia or astrocyte cultures or tricultures together. So that's the kind of experiment we're running at the moment. We're actually expecting a LISA result any day. So it would be fun to see whether our hypothesis check out in this model, and whether the triculture model performs better than monoculture of neurons as well.
Okay. I think that may tie in with one of the last questions that we got in the comments. So as you were mentioning with those with the triculture models, then have we tried or potentially looked to design these cells or assays into organoids?
We haven't. However, there are companies that have taken our cells and have built organoids by themselves. And if you reach out to us, we'll be able to continue this conversation.
Yeah, I agree. There's definitely been groups that have used the organoids. For those who may have some interest, if you're able to email us or share your email, there are some publications that have used our differentiated cells to make organoids and 3D tissues as well. So if you are interested, please feel free to reach out. And then I think that is it. So yeah, so anyone from the emailed questions or in the chat, please know to make sure that your email is correct. We will send an email out to answer your questions. And with that, I'd like to move into this last little point after this Q&A, that as you've heard from our great senior director of R&D, her team, as well as many of our other team members in different departments, are some of the greatest strengths we have here at Elixirgen Scientific in regards to the great science that they do and the want and need to be able to help all researchers out there reach their goals. So we try to offer more of a partnership solution approach that allows us to carry through not only for these 80 panel assays for more of a translational research approach, but we also have capabilities based off of our core technologies to help with generating new cell types. As you saw, we can generate many cell types. We just need to have the request for them, and we do partner in that sense. If you're in need of a large amount of cells to run more high throughput, we offer differentiated cell supply partnerships to help make sure you can have those types of cells at the high scale as needed. As well as we've been branching into iPSC-derived cell therapies. or if any of these are of interest to anyone here, please feel free to reach out to us. We're more than happy to discuss any of these approaches, because our main goal here is to make sure everyone here succeeds in the research that they're trying to get done moving forward. Then next slide. So for those in the Boston area, if you are going to be attending the Alzheimer's and Parkinson's Drug Development Summit next week at the Riviera Hotel in the Boston Common area, we will actually be in the Boston area all next week, including Nadia at this event. If you are interested in discussing either at this event or potentially meeting in the Boston area during next week, please feel free to reach out to us, and we'd be more than happy to set up a face-to-face and get to learn more about your research, as well as share some of the great advancements, including some of our advancements towards the Parkinson's drug development process as well.
Okay, I'll add some Parkinson's data.
Yep. And then for those who may be interested in wanting to get a quick jump start into this Alzheimer's disease panel service, this is just a quick layout of what we have just currently available that we can get started as technically as early as next week. So if this is of interest, this is just a quick lineup, and please feel free to email us at our info@elixirgensci.com, which is then on our next slide. Just as part of our things, you can see you have the email at the bottom. Please feel free to reach out to us there. And with that, thank you again, Nadia, for that wonderful talk. And thank you to everyone who attended. And hope you all have a great rest of your day and a wonderful rest of your week.
Yep, and folks, if you have questions about this talk or any data that I've presented, I'm on LinkedIn. There's email. You can reach out to Henry. So let's continue discussion.
Sounds perfect. Thank you all
Awesome.
again. Have a great rest of your day.
See you.